fun. Why not? Okay, so we're gonna finish up our cytoskeleton before moving into cell cycle. Everyone got their papers, just in case I decide to give any stickers out? So if you would like some, take it.
Yes, can I have one?
Eventually it will come up, but just to remind you, last week or last class, we were talking about different cytoskeletal structures, right? And up till now, we've talked about intermediate filaments, we've talked about microtubules, and for each one of them, we've looked at how they're built, right? What type of things they do, functions that they are involved in. So your intermediate filaments, it was the same amino acid chain, same polypeptide chain, that kind of does a coil around each other, right? That's a single active unit of that. And then they can make octamers, tetramers, whatever, to build thicker fibers, thicker bundles, right? And they'll rope-like structures. Your microtubules, on the other hand, are hollow tubes, right? That was a big difference between those two. So your microtubules were hollow tubes, and they were built of alpha and beta subunits, right, combined together. They were driven through GTP, right? So there were GTP bound monomers that would come in to build, and then hydrolysis of that would lead to the destruction. And they, you can have polymerization to make them elongated, and then depolymerization to make them shrink and go away. Right, and that the end where more molecules are coming in, the GTP tubulin is coming in, that end is your plus end, right? That's the one that's growing. And then the opposite side, which does not have the GTP cap, so doesn't have all these GTP tubulins stuck next to each other, is your minus end, and that's where typically they are going to be going away. So when they start to shrink, it's because they lose the GTP cap, right? Because the rate at which they're coming in is slower than the rate at which they're losing them. And that leads to then their shrinkage. You can restart the same end as a plus end if you can reestablish the GTP cap. Again, it's just a balance between how quickly they're coming in versus they are losing it. They have, which means that they're dynamically unstable. You can fix them, right? You can make them stabilized by connecting them to a cap or by connecting them to specific proteins that stabilize them during specific functions that the cell uses them for. Actin is the last one that we started to talk about. Now, actin also builds very similarly to a tubulin polymer, right? Except in this case, actin filament is formed with the actin monomer coming in, and it uses ATP hydrolysis to build and to get out, right? So actin bound with ATP is what comes in at the growing end, and actin with the hydrolysis of ATP, the ADP bound actin is what's going to be lost to shrink those tubes. And then this one, in this case, they can be filaments, right? They don't have to be hollow. These are just, again, rope-like structures, right? So the microtubules were the only ones that were hollow in the center. The other two are filamentous rope-like structures of various types. In this case, this is made up of all these globular actin monomers stuck together to create that rope-like structures. There are many ways in which they can be utilized inside the cell, and many ways that they can be structured inside the cells with the help of linking proteins and binding proteins that they interact with. So you have nucleating proteins that are going to help increase their length and help to grow them. You can have severing proteins that can cut them up into smaller pieces as needed for specific functions. You have cross-linking proteins. These are especially important in the cell cortex, where they are used to keep that scaffolding structure in place. You can have capping proteins that are going to block the growing end and stabilize them, so they no longer grow or shrink at that point, right? You can have site binding protein like in tropomyosin or in myosin motor protein that can help them move in different ways to help the muscles contract and relax. And then you can have bundling protein that can help them build stronger structures that are usually part of movement systems, right? So in this case, this is for filopodia, but you can also have similar structures in other such movement sites. There are, just like there are drugs that can target microtubules, there are drugs that can target actin filaments, and those can then be detrimental to cells and lead to cell death eventually. The biggest one that we typically use are phalloidin, which bind and stabilize these filaments so they cannot move any further, right? It kind of freezes them again in time. So if they wanted to do cell cycle, they can't because they're stuck. They can't move, they can't do anything, and eventually will die out. Other two drugs that are important are the cytochalasin and latrocinidin. And these two, they are going to prevent polymerization in different ways, so they cannot build to begin with. So they're stuck and the cell will collapse, essentially. Okay, so the biggest function of this that I want you to remember, there are other functions, obviously important ones too, but a very important function of this is in the cell cortex to maintain the cell scaffolding and cell shape through that. And in this structure, you have your actin filaments, right, that are going to be all around right underneath your plasma membrane. They are bound together with a complex of binding proteins. You have spectrin dimers. It's another filamentous protein. These are thinner proteins, right, that work together as a dimer, and they link different actin monomers together. The spectrin dimers interact with other attachment proteins that can help at the cross-linking sections to keep them in place and to keep that scaffolding stronger in those places. The actin fibers can directly bind to the attachment proteins as well and can function through that, but many times they function through indirect binding with the spectrin dimers instead. Another important function with actin is movement of the cell. And this is just a simple cell moving with like their little protrusions with the plasma membrane, right? So these lamellipodium or any type of like phagocytosis, right? In white blood cells, that's dependent on these actin fibers as well. So you have your cortex structure, right? All underneath your plasma membrane, that's going to be all made with actin. So when you have a need for movement for whatever reason, there will be signaling molecules that will cause actin polymerization at the site where they need to move, in the direction that they need to move. Particular fibers become the growing end of the actin fibers at that point. So the area which has the activated plus ends, that's the direction in which the cell is going to end up moving. And so, as these grow, it causes tension on the remaining of the cell, causing it to move towards the site where the actin fibers are moving and moving it in that direction. So it keeps kind of moving it towards that site. Part of it is also going to include myosin motor proteins that are going to help the back end of the negative side, right? The minus end of those actin fibers in contracting and moving along with the side that is moving forward. Okay, so these, again, like I said, these are the actin fibers, and that are going to lead this movement by having the leading edge of the cell is where all your growing actin monomers are going to be. So this is where the ATP actin monomers are coming in, adding on to that site and growing. And when they grow these things, that's going to make that long protrusion that you're going to be looking at. In the back, you will have obviously the negative side of the actin filament. So they will be depolymerizing there'll be some molecules coming off. It's just that they'll be polymerizing at a much faster rate in the front than they are depolymerizing at the back. And in the rest of the region, you will have your actin fibers stabilized with the help of the capping proteins. So in this region, no change is happening. In the front end, they are moving, they are growing, and in the back end, they are depolymerizing. So that's what's going to cause them to move forward towards that site. So how does this happen? This happens through signaling molecules, as always. And these can be, yes.
I was just gonna ask if you can dim the lights again, a little bit.
Yeah, you know how about we try to dim lights in the back? These don't listen to me. In the front.
It's just really hard to see.
I know, it is, and I completely, is that good?
It's better.
Yes, we will, I know, now everyone's gonna go to sleep. It's me on this stuff. Hopefully not. Anyway, but just like any other thing, it's signaling molecules that the cells are using to get signals from outside, right? Typically you're moving because there's too much light or something has happened, right? You touch something odd, whatever it is. So it's an external signal that's going to make you move in most cases. So those extracellular signals are getting received by the cell membrane, and they are going to alter the actin filaments in that region when they are activated, right? So in that leading edge, the reason that became the leading edge was because that's where they received the signal, and that's how they shifted. And so there are a few different signaling molecules that can activate cells to behave differently, to move in different ways. So here is a typical unstimulated cell. It's just kind of, spread out and hanging out in the plate or in the body exactly the way it needs to be, with no leading or lagging edge. But then if you, for example, if there is a protein called Rho, if you have Rho activation of signals, the area where the Rho receptors were bound, where you have the Rho activation, will become the leading edge. And that's the direction in which the cells will move. So all the actin fibers are going to start, all this edge is going to have those growing ends. This side is going to be depolymerizing. So the cell will move in that direction where the Rho activation occurred. Another type of activation that can happen is CDC42. This is a different type of signal that will cause the cell that maybe there is something toxic in the environment that might be switching it on, and it will cause the cell to completely try to have minimal interaction with its environment. So they ball up and they become rounded up structures. So they're not really moving in a particular direction. They're trying to minimize their interaction with whatever toxic is there. So they will round up and become for that. And then other things that can happen is RAC activation, where maybe whatever is in the environment requires them to have further binding to the extracellular matrix to have a better sitting there, better response. And so in that case, they become even broader and they become even more established with the environment. Other places where it is important in the way it changes the shape is cell cycle. This is how the cells divide into two, is through this ring made of contractile ring at the side point of cytokinesis that is built of actin and myosin filaments combined together in that cleavage furrow. So you have your interpolar microtubules. If you remember, they were the ones that were those stable interactions in the center, right, at that metaphase plate. And then they were the ones that continued to grow outward so that you were elongating that cell as well. So these stable interactions in the center, which was holding them in place, is going to mark the space where the contractile ring of actin will form. And then eventually lead to the separation of the two cells. And here you can see this in an electron micrograph where they are the actin and myosin filaments stained to show you where they are. and they weren't present at this time. There is no actin filament present anywhere else, right? Because the cells are just beginning to form the new cells. So just going over the actin itself, actin filaments are, again, rope-like structures. They're very thin and flexible because remember, they're made of these ring globular proteins, subunits, right? So that makes them inherently a lot more flexible than something that has more form, more lock and key type mechanism. Both actin and tubulin polymerize by the same mechanism or very similar mechanism. Tubulin with the GTP bound tubulin and actin with ATP bound actin monomers. Many different proteins are going to bind to actin, and depending on what proteins are binding to actin, it will have different functions, right? So if it's binding to spectrin, it's going to be part of the cortex. If it's binding to another protein like CDC42 or another thing, then it's going to have functions in regulation, right, or movement. So depending on what it binds to, it will have different functions. The cell crawling mechanism depends on this cortex actin that is getting influenced by whatever signaling signals, extracellular signals it receives. And finally, actin can associate with myosin to create contraction and relaxation response, which is part of how cell cycle, in cell cycle, you get that division of cells in cytokinesis. So muscle contraction, like I said, I do have that little video online as well, but I'll just quickly go over a little bit and move on to cell cycle. So in your muscle contraction or any type of contractile ability that you see, which involves actin, actin doesn't work alone. Actin works in conjunction with another filamentous protein called myosin. A single myosin is made up of a rope-like tail with two, these globular heads. So a simple myosin protein, this is again in its quaternary structure, is made up of two monomers of myosin combined together, each one containing this head and a tail, right? The filamentous tail that coil around each other, making this exact structure. Now, in a myotubule or in a muscle fiber, what you will have are bundled these myosin structures, where you have these myosin tails in the central region of the myosin tails interacting with each other, bless you. And then you have all these filamentous heads facing the outside and all the tails towards the inside. And they are going to be laying against each other on the two sides so that you have both ends of the filament having the myosin heads, and in the center, you just have the bare regions of the tails. The actin fibers lie next to these myosin filaments, and the actin fibers will have their plus end towards the globular side and their minus ends towards the tail side. So remember, the minus end is where they're depolymerizing, right? And they're shrinking, and the plus end is where they're extending. And so they would be stretching essentially these myosin filaments outward or inward. And that's where you get that contractile motion happening. So in a muscle, right? If you look at a single unit of a muscle fiber, those are these sarcomere units, right? They're about 2.5 micrometer in width. And so you have these overlap regions in the center, right? And if we were to take a cross section of it and look at it further, they are made, so our muscles are made of these myofibrils, bundles of these myofibrils, right? Each one of these myofibril is essentially made of these bundles of myosins and actin filaments lined up next to each other. So here you have your myosin filaments, right? These are those little bundles that we just talked about with their globular heads on either side. And this central region, this is just the tail sticking to them. And then you have these thick actin filaments on either side, okay? So the thick actin filaments are anchored at the C disc. This is where they're going to be kind of hanging out together. That's gonna limit how much they can extend and contract. So any muscle, especially, in this case, we're showing a skeletal muscle, is gonna be packed of these myofibrils. made up of your thick myosin filaments and then your thin actin filaments to either side. So when they are working, what they are doing is your plus or the growing end of your actin is going to be increasing or polymerizing, causing it to extend. And when they stretch out, you have just your muscles. They are in the fully relaxed state, right? That's going to be the highest they can go, the longest they can extend. And you'll have minimal interaction with the myosin heads. As they contract, as you lose those fibers at the minus end, right? At the actin become shortened on the minus end, they become closer to each other, and you have extensive extensive interaction between the actin and the myosin heads. And that's going to be when they're fully contracted. In their full contraction, you will have all the myosin globular heads interacting with actins on either side with no free myosin head available. And then in the extended form, you will have just a little bit doing it. How does this happen? Is through more proteins as well, always extra binding proteins in there. This is also going to use ATP again in that process. So here you have your the tail region, you have a lever arm structure, and then the myosin head that you're looking at. It's going to be interacting directly with the actin filament in some of the actin globules, right? So those individual monomers. ATP, when it's taken in, is going to activate this myosin filament. So when the ATP is bound to the myosin globular head, it's no longer able to bind to the actin, and it's going to get freed up, and that's going to cause it to move, right? And the actin filament to slide away from it. And this process essentially hydrolyzes ATP, right? So it has the ATP bound to it. It can't touch actin anymore. It's going to hydrolyze it. And that hydrolyzation is going to cause it to shift forward. Now you have the ATP myosin. It can bind to the next actin on the filament. And this can continue on until it gets all the way to the last one. Yes.
So does the myosin want to contract by default if it's not connected to the actin filament, or is it the opposite of that?
So this ATP bound myosin is not stable. It will get hydrolyzed. So when it gets hydrolyzed, it automatically then shifts it inward. So that would be its resting state, would be in a more contracted state than relaxed.
So the rested state's actually the contracted state, not the
Yes.
Oh, that's interesting. That's the opposite of what I expect.
So well, for the relaxed state is where you have minimal interaction with the myosin heads. So you have to remember it that way. So as a whole, the relaxed state is going to be your resting state, is going to be your least energy consumption. But once activated, the way that it will move is in the contracted state. That is the only way that it can lose that energy that was put on there that's unstable.
Okay, this is gonna be weird, but ATP is like this, ADP is like this.
Yes,
Okay.
exactly.
Yeah, that's all I need to know. Thank you.
So yes, but what is happening is that that's causing that, when the ATP is hydrolyzed, it's shifting it inward to the next actin head. And that's what's causing the contraction to happen as well. Okay, so at the level of yes.
Sorry, one more question. So it's not the myosin head that pushes it forward. It's just that when it
Hydrolyzes. It's because the ATP bound myosin
Yeah.
is no longer able to attach to actin,
Okay,
right? So when it hydrolyzes, that energy is what's pushing it forward.
I see.
Right? It's not that the myosin is shifting it forward. It's that hydrolysis the happening of the hydrolysis is releasing that phosphate, and that's causing a power stroke in the myosin head so that it can now bind to the next actin over.
Thank you.
Okay, so what exactly causes this is an influx. Obviously, it has to receive some signal that will cause it to relax or, contract. So the contraction actually happens with the sudden influx of cytosolic calcium or sudden rise in that calcium. So you have your lumen of the T tubule. This is your extracellular space, right? And then you have lumen of the sarcoplasmic reticulum. That's going to be in your muscle fiber. And you have all this calcium that's built in one side and the other. When the cell receives they are connected to each other through a voltage gated calcium channel on the lumen side in the extracellular space, and a calcium release channel in the sarcoplasmic reticulum, in the actual muscle fiber. So when an action potential comes in, so when there's a signal coming in from a nerve fiber causing, asking you to push in or pull up or whatever the function that needs to happen from the muscle, it will directly impact this calcium release channel and release all the calcium from your sarcoplasmic reticulum, from that inside the cell, outside. And when it releases it, that's what causes the contraction of your muscle fiber. So it's not influx of it inside, but rather release of the calcium from the muscle into the outside environment. Does that make sense? And then obviously it will need to then come back to a resting state, which is done through this activated T tubule releasing its calcium and then bringing it back to a homeostatic state or resting state. So here you have a skeletal muscle that has your actin fibers, and then you have tropomyosin fibers that are wrapped around it. Again, these are going to be wrapped along the sides of it. In the first one, the myofibrils were sitting next to each other, right, in the sarcomere. But now in this particular skeletal muscle, these fibers are wrapped around the actin. And you have these troponin complexes. These are helper proteins that are keeping the actin and the tropomyosin together in those spaces. They're kind of anchoring them all along the way. So typically the tropomyosin is going to be blocking your myosin binding sites in your actin. So this is your actin, the tropomyosin, this fiber that's wound around the actin, it is keeping it closed. When calcium comes in, right, when you have that action potential, calcium comes in, it causes this tropomyosin to shift off the actin. So these to come off. This releases this site where the myosin globular head can come in and bind. Okay. So they're all working together, right? So we were looking at those individual fibers. We were looking at myosin and the actin. Well, along the actin, you have these tropomyosin heads, or not tropomyosin filaments, that are keeping it from binding to your myosin globular heads unnecessarily, right? So they will only bind to it when there is need for the contraction and for this process to happen. So you have your blocking sites that are keeping it together so that myosin heads don't bind. Calcium comes in, it takes away the tropomyosin. So now the globular head can bind to it. ATP comes in, that is going to activate the myosin, so it will come off, right? And then it's going to get hydrolyzed and bind to the next one over, and cause that contraction to happen. And then once that process is finished, the calcium-mediated trypomyosin movement goes back when there's no more calcium, extra calcium around, right? All that calcium that was released comes back into the cell that is going to close that up again so that it can no longer keep that activated, keep that process activated. Make sense? Okay, so that's basically the muscle contraction process. So again, around these, what is not shown here are your tropomyosin filaments that are keeping this process from happening normally until they are activated by this calcium release. Yeah. Okay. So that's it. No, any questions on this part? Yes, no, maybe? Whoa.